Welcome everybody to today's Latinx Abroad speaker series. My name is Rafael Sanchez. I'm one of the co-chairs of the Latinx Abroad Steering Committee. And before today's presentation, I would like to talk to you about Latinx Abroad. So the Latinx Abroad group is an affinity subgroup at the Broad Institute with a mission of bringing people together and empowering them to reach their full potential. We have a dual, with the speaker series, following this mission, we created the Latinx speaker series with the purpose of highlighting Latinx and diverse researchers within and outside of the Broad, fostering collaboration, and really empowering people to build connections and have a better understanding of the research environment within the Latinx community. So with this in mind, I would like to present today's agenda. We will be welcoming Carlos Eduardo Guerra, who will be presenting on an evolutionary perspective on human disease and adaptation. Just before the presentation, I would like to give a brief intro, share a brief bio of Dr. Amorim. Dr. Amorim received his bachelor's and master's in science from the Universidade de Brasilia in 2006 and 2009, respectively. He received his PhD in 2013 from the Universidade Federal do Rio Grande do Sul. He moved to New York and did his postdoctoral research in the Tversky lab at Columbia University. He is currently an assistant professor at California State University, Northridge, and is also a population geneticist interested in human evolution, anthropology, medical genetics, and leveraging large data sets to contribute to the understanding of the biology of our species and of its closest relatives. He would like to understand the interplay between demography, mutation, natural selection, and culture in determining the patterns of genetic diversity of human populations and how it impacts human health and evolution. He has experience of genomics, forensic genetics, public health, and anthropological genetics. He has worked with African and Brazilian communities and indigenous populations, and lately has been working with large public genomic databases from humans and other species. Welcome, Dr. Guerra Amorim. The stage is yours.
So thanks everyone for coming, and thanks for the introduction. My name is Eduardo Amorim, and I'm an assistant professor at Cal State Northridge, and I'm very honored to be here giving this talk at the Shades Latinx Speaker Series. You might have noticed that the title of the talk is a little bit different from what it was initially in the website. Let me just see one thing here. Okay. On the website, but I'm going to be talking about more broadly about different projects that I've been doing in the past few years. And the title of the talk is An Evolutionary Perspective on Human Disease and Adaptation. So before talking about science specifically, I want to talk a little bit about my history, where I come from, and where I am now. So I go by Eduardo Amorim, as you know, and I use male pronouns, he and his. I was born in Brasilia, which is the capital of Brazil, and here on the left you can see some photos of Brasilia. I want to talk about my trajectory because I don't know many other people. I actually don't know one person that has a story like mine that came from Brazil or Southern American country, and is currently and did the PhD there and studied there and moved here and is now an assistant professor. So I did my PhD and master's in Brasilia, where I was born, and then I moved to Porto Alegre to do my PhD. I then came to the US in 2014. I did a series of postdocs. I moved to Switzerland in 2020, and then I did another postdoc there. And more recently, in August 2021, I started as an assistant professor at Cal State Northridge. So this is all for my story. I also want to talk a little bit about Cal State University system. We are 23 campuses spread all over the state of California. And Northridge is down here, and it's located in LA. I'm going to show where in a bit. And since we're talking about Latinx and shades, I also want to show a little bit the profile of our students. Most of our students are Latinx or Hispanic, and we have also another diversity of many other ethnicities and races. This is my team. We are 4 currently. I'm the PI, and I have a postdoc that started this week. And there are 2 master's students that are working with us. They are officially joining in August. And on the bottom left, you see here a photo of California State Northridge. You can see the mountains in the back. It's very beautiful. And on the right, bottom right, you see the map of LA County, showing that we are here in the valley in this top part here of the map. So as was mentioned in the introduction, I'm a population geneticist. So we in population genetics, amongst other things, we are concerned about understanding the causes and consequences of genetic variation across individuals and populations. And we studied different genetic processes and evolutionary processes. I specifically focus on some evolutionary forces such as mutation, genetic drift, natural selection, and gene flow or admixture between human populations. And my specific interest is to use genetic diversity and study genetic diversity to understand how evolutionary forces shape our health. And today I'm prepared to talk about 3 different projects that I've done in the past few years. The first one is concerned about finding the determinants of the frequencies of disease mutations in human populations. So in other words, why some human populations carry a larger burden of disease mutations than others. The second research project that I'm going to talk about is what determines the selective effect of a new mutation. So when a mutation arises in the genome, what determines if that mutation is going to be neutral or beneficial or deleterious, for example. And the last project that I'm going to talk about is something that I'm working right now. I don't have finalized data yet. I have some preliminary data, but this third project is concerned with the question of how did humans adapt to extreme environmental conditions. And here in my research, I'm focusing on hypoxia, which is low oxygen levels that is typical of high altitude environments, and also ancient epidemics. Okay, so going to the first project. This project, as I mentioned before, is to understand the determinants of the allele frequencies of disease mutations in humans. This is actually the very first work that I did when I came to the US, so it's a little bit old. We published in 2017, but I wanted to bring that today just to contextualize the type of questions that I ask, trying to use evolutionary theory to understand a human disease. Okay, so the main question, as I mentioned before many times, is to understand what determines the frequencies of disease mutations. And this is the paper. And in this paper, we use this type of analytical models that explain, for example, what's the frequency of disease mutation, and it's determined by the mutation rate and the population size, for example. One way to, sometimes it's easier to understand with like small cartoons. I'm going to show here what I mean with this type of models. So here we have a sink. We have like the tap that's putting water in the sink, and then we have the drain that it's eliminating the water from the sink. So the sink would be the population. We have the tap that it's the new mutations that are coming in, and natural selection is purging these deleterious mutations, which is the drain. in the bottom of the sink. So if we increase the mutation rate, so we put more water, you're going to have a higher frequency of disease mutations in the population. And if you increase the strength of natural selection, so you purge deleterious mutations more efficiently, you have a lower frequency of the disease mutations in the population. So this seems pretty simple, and actually it helps us understand a lot of disease evolution in humans. But the fact is that our history, our demographic history is very complex. We have a lot of episodes of admixture, gene flow between populations, right? We have population size changes across time. So those analytical models sometimes don't capture all the complexity of our population history. To address this type of more complex questions in human evolution, what we do is that we use these analytical models, but in the framework of simulation. So we simulate genetic diversity following those basic rules that I showed in the model. And then we can contrast the simulated data versus large-scale biomedical data sets. In the time that we were doing this work, we had previous versions of the NOMAD data set, which was called XAC, which was focused on the exomes. And we used that in comparison to simulations to understand what were the determinants of disease mutations in humans. One of the findings of that paper was that in general, there is an excess of deleterious variants, actually of lethal mutations in human populations worldwide. When I say lethal, I mean specifically recessive lethal. That was the topic of the paper. So we found an excess of those. So the simulation showed that we would expect to find a lower frequency, and the real data actually has a ton more than the expectation. And we have different results in the paper. This one is the one where we compare the expectation for each gene versus the real data. And here on the x-axis, I have all the list of genes that I analyzed. All those genes, mutations on those genes can cause recessive lethal disease. And the gray bars and the gray violins, they show the expected allele frequencies of disease mutations in that gene over 1 or 2 million simulations, a very large number of simulations. And the bars in the bottom show how many times the simulation showed no deleterious variation. And you can see here on the leftmost gene, the CFTR, that the simulation showed that CFTR should always have some sort of disease mutation, for example. We then compare these gray distributions with the observed allele frequencies in this data set that I mentioned before, and those are the purple bars. And what you can see here is that the purple bars are usually on the top of this distribution, so it's in the extreme of this distribution. And we find that a few genes, those 7 genes here, are the ones that present an excess of deleterious variants as in comparison to the expected. Those deleterious variants, as I mentioned before, those are recessive lethal. So we were trying to investigate what is actually driving this excess. In the paper, we analyze a few factors. Here's a list of different factors. One of them is treatment, for example. We know that cystic fibrosis, for example, that it's involved with the CFTR gene. There is treatment for that. So we also simulate treatment, for example, and we do a lot of different adjustments in our models and simulations to see if those factors could explain this discrepancy that we observe. We actually, the ones that we were able to evaluate, they couldn't explain the excess. So there's still a question that we couldn't answer, which is really what is driving at least some of the genes that we analyze, driving this excess of deleterious mutations in those genes. One potential, one possibility that we think that could be doing this is the misspecification of the selection coefficient, meaning we are assuming the selection coefficient is 1 and it's actually something else. We know that those mutations are recessive lethal, so we expect a selection coefficient of 1, but there might be different in the past. So with this question in mind, like, can selection coefficients change through time? I developed a second project that I'm going to talk to you right now, and for which I have some data. So here is the main questions. So why certain populations harbor an excess of deleterious variants, and what the term is the selective effect of mutations. So can the environment have some influence on whether a mutation is going to be more or less deleterious, for instance? So this is the motivation to go to the second project, which is a project that analyzed wolves and dogs. And just one second. So in this project, we analyzed this selection coefficients as the distribution of fitness effects, which is this DFE. So the DFE describes the distribution of S, the selection coefficients, in the genome or part of the genome. In other words, it quantifies the amount of deleterious, neutral, and adaptive mutations that enter a population each time. And this DFE can inform us about the ability of an organism to adapt to its environment, and also how disease mutations can spread in the population. So in that sense, it relates to the previous project that I was discussing before. And this is one DFE. This is not from my paper, it's from a colleague, just to show you what I mean with the DFE. You see for each species, we see human in blue and Drosophila in red. For each species, we see 4 bars. And each bar corresponds to a specific strength of natural selection that we see on the x-axis. So we have on the left the neutral or nearly neutral mutations, and on the right we see the strongly deleterious and lethal mutations. So here what we can see is that humans have, for example, a larger proportion of strongly deleterious mutations in their genome than Drosophila. And there are different ways that we can study this DFE. There are experimental approaches that people are doing with bacteria and also with Drosophila. And there are methods that are based in polymorphism data that we can use, for example, to study the DFE in humans and other species for which we cannot do some experimental approaches. So my approach, my way to study the DFE in dogs and wolves is with this method that was developed by my colleague, Bernard Kim. And this method, it's based on the analysis of SFS, so the site frequency spectrum. We use synonymous mutations to infer the demography of the population that we're studying, and also to control for the effects of background selection, which is the selection on linked sites. We use this synonymous SFS to infer demography and background selection effects, and we control for that, and then we infer the DFE using the non-synonymous mutation. So roughly speaking, if non-synonymous mutations were all neutral, so all mutations are neutral in the genome, we would expect both SFSs, the synonymous and non-synonymous, to be the same. Also considering that the mutation rates between synonymous and non-synonymous is the same. So any departure from this can be attributed to natural selection on target sites, which is what we are interested. So this framework is developed into this method called DADDY and FitDADDY. And that's the results that I'm going to show for you now. Before showing the results, actually, let me contextualize what I'm trying to analyze here. So I mentioned before that I was interested to understand what determines the selection strength, right? So why a mutation would be strongly deleterious or neutral and or beneficial. This question has been asked by many other groups as well. And this is one field of research in population genetics, which is the DFE evolution. One of the first works that used that inferred DFE in humans and in Drosophila is this one that I already showed before. They also analyzed yeast and mice, and they find that as species become more complex, the proportion of deleterious mutations in their genome increase. So there is, they associate the proportion of deleterious mutations with the complexity of the organism. And the other work that follows this, and it's more recent, is this one that analyzed the DFE in related populations. So they took 2 human populations, 2 fly populations, and 2 tomato populations, and they checked, they evaluated if the DFE were similar between these populations. What they see is that the DFE is very similar, but it's more different as the species are more divergent. So for the populations of tomato, for example, they were more diverged than the other ones. Their DFE is less correlated. So we wanted to use this type of approaches to understand how the DFE can change across population and how fast it could change. So we used dog as a model system, dogs and wolves and a model system to study rapid and strong natural selection shifts. We chose dogs and wolves because they are very similar organisms. They have very similar genomes, but they're subject to very different selection regimes. One is subject to artificial selection. We know many of the traits that we selected for, and the other organism is not subject to artificial selection. So for the reason we think dog and wolf would be a nice system to study this type of questions. In our study, we have 4 different dog breeds. We have Labrador, Tibetan Mastiff, Border Collie, and Pug. We also have a population of Arctic wolves. Sample sizes vary from 6 to 20 individuals. So that's 12 to 40 chromosomes. Coverage is around 30x, except for Tibetan Mastiff, which is lower coverage. And we, in addition to these populations, we also add some artificial populations using dogs and wolves from lower coverage. When I mean artificial is that we are lumping together dogs that are from different breeds into one single population. And we do the same with the wolves. We get wolves from different populations and we merge them together into an artificial population. So we have the different settings. And those are the SFSs that we calculate for each one of the populations. We can see the red bars representing the synonymous mutations, and the red bars, sorry, the green bars, the synonymous mutations, and the red bars, the non-synonymous mutations. And on the left, we have the SFS based on the count of these mutations in the population. On the right is the proportion. So one thing that we can see here is that we see more synonymous mutations in number than non-synonymous mutations. We see that on the left. And when we look at the right, we see that the non-synonymous mutations in red, they are usually found in lower frequency than synonymous mutations. So this is what we expect if there is natural selection going on. And when we apply the methods that I mentioned before, to infer the DFE, what we find is that the DFE is stable across canids. So that means that the DFE is not changing from dogs to wolves. Here I have one of our results. It's the same thing as I showed before, the DFE, right? So you have for each population, let's look at the AW, which is the article, for example, is the yellow bar. We see 4 yellow bars. The left one shows the proportion of neutral or nearly neutral mutations, which in the article is around 32%. And on the right side, the last bar on the right-hand side, we see the proportion of mutations that are lethal or nearly lethal. And in the article, for example, is around 47%. So what we see here is the results for all the populations we analyzed. We have the dogs in blue, the wolves in yellow. And we don't see any difference when we look at specific depth of coverage. So the first 4 bars in each group of bars are the high coverage genomes, and the last 3 are the low coverage genomes. So the only differences we see here that are significant are the ones related to the depth of coverage. We were puzzled with that, but then we realized that when you have missing data, like in lower coverage sequences, this missing data is not evenly distributed in the genome. So it more often happens in sites that are under weak selection or that are neutral. So this is the reason that we see the shift. But when you look at different like data sets with the same with similar coverage, you see that the DFE is very similar. So this result showing that the DFE between dogs and wolves is very similar is very consistent when you use when we assume different parameters for in our analysis. For example, different mutation rates estimated by different authors. Also, the non-synonymous to synonymous ratio, mutations or like sequence length. I calculated that myself for dogs, but we also consider the same ratio for different organisms and see how it behaves, our analysis behaves. Also, we assume these different distributions for S. These results here on the left show a mixed distribution of neutral, a mass neutral, and then a gamma distribution. But we assume also like log normal distribution and a gamma distribution for instance. And everything always shows that DFE in dogs and wolves is very similar. So the conclusions for based on these results, the conclusions that we have is that there is apparent no impact of domestication or breed formation or artificial selection on the DFE, at least not in a genome-wide scale. So this stability of the DFE, despite artificial selection, suggests that the DFE, at least in dogs, is determined by organism characteristics with little or no influence of the environment. So even the environment changing a lot, at least in dogs, we don't see this shift in the selection coefficients as we initially expected. When we extrapolate these results to the human recessive lethal mutations study that I mentioned before, and we did see some discrepancies in our analysis from in the simulations relative to the data. So we initially thought that could be some differences in the selection coefficient over time. At least based on this analysis with the dogs, what we hypothesize is that probably that was not the case. Probably selection coefficients are very stable over time, especially if it's negative purifying selection. So with that, I want to talk about my last topic for today, which is this project where I use ancient DNA to understand human recent evolution and also resistance to pathogens. This follows from the previous project in the sense that I'm still interested to see how natural selection changes genetic diversity. And in this case, I'm using populations from the past. I'm using ancient DNA to see how natural selection was in the past and comparing that to present day populations, for instance. And the results that I brought here are very preliminary, but they are mostly focused on populations living in the Andes. The Andes are these mountains in the west of South America. It includes many countries like from Venezuela, Colombia, Peru, Ecuador, Chile, Bolivia, and Argentina. So many, many countries, almost all countries in South America. And it has this city called Machu Picchu, which is a very famous archaeological site. Many people know that because it's a very famous touristic attraction as well. And the curious thing about this is, well, the And in the Andes, throughout all the Andes, there are 80 million people living there. And why this is surprising, it's because the Andes are located in high altitude. So people living in the Andes are living at 3,000 to 7,000 meters above the sea level. And above 2,500 meters above the sea level, the oxygen concentrations drop to 70% relative to that at the sea level. And this 30% less oxygen in the air causes a lot of different diseases, a lot of different symptoms in humans. Here's a list of these different symptoms. The very like the well-known one is preeclampsia, but all of them are related, or most of them are related to phenomena that we think are related to natural selection. So we think living in high altitude should be an example of living in an extreme condition because of this several challenges that the environment imposes. And nonetheless, although we see all those challenges, humans have peopled high altitude adaptations for a very long time and in many different places of the world. Here are the three examples that we know. Tibetan Plateau, which is the Himalayas. There is one in Africa, which is the Ethiopian highlands, and the Andes that I mentioned before, which is in South America. So our results related to this research are still preliminary, as I mentioned. I want to show you a little bit of the data that we have generated, and then I'm going to tell you a little bit about the population structure that we detect, and show what are we doing for influence of natural selection. So this is the data summary. We have genomic data, whole genome sequences for 73 individuals. The mean depth of coverage is very low. It's 1.6x. And you see that it ranges from 0.01x to 7x. This is pretty low in human genomic studies, but it's common in ancient DNA studies. And that's because DNA degrades over time. Our individuals that we're analyzing, they lived 500 years ago, just before the contact of Native Americans with Europeans, up to 9,000 years ago. And we get those samples, 73 samples from different ages, and we extract DNA from those. Mostly we have teeth, but we also have pieces of the skull that have a lot of DNA. And using specific techniques and specific, like special laboratories, we extract DNA from those. And I'll show what type of data we get from that. But just again, talking a little bit about the sample here, like 2/3 of our samples come from high altitude populations and 1/3 from the coastal populations. Here's the distribution. Each black dot here is one sampling location. The sample sizes are indicated in the figure. And we have samples from Colombia, we have samples from Peru, Bolivia, and Chile. So, okay, so when we analyze ancient DNA, we sometimes don't get the DNA from the species that we actually want to study, in this case, humans. Here I'm showing how much human DNA we get for each sample, which is the blue bar. The orange bar shows the microbe DNA. It can be ancient pathogens, but most of it are actually microbes from the environment. And here you see that some are very low, like 1% or less, and some are very high. This is not a big problem. Of course, it would be ideal that we have a lot of human DNA in each sample, but that's just the nature of an ancient DNA sample. Another thing that, okay, so when we, so we get this data, we separate the microbe DNA from human DNA, and then we check this human DNA to see if they have this characteristic pattern of ancient DNA, which is an excess of C to T transition towards the ends of the reads. I'm not going to go into detail, but I just want to say that we authenticated our samples as ancient DNA samples. With this data, what we do, sorry, checking the chat. Okay. With this data, what we do is that we, first thing that we do is to check for population structure, just to see if we can actually consider these individuals as one single population for further analysis for natural selection. And commonly what we do is this type of MDS plot or PCA plot. This one is an MDS, and each point here corresponds to one individual. So like we have the red dot and the other red dot here that are very close together. And as closer they are, the closer they are genetically as well. So this is the physical distance in this plot is proportional to the genetic distance. And what we see here is a clear structure relative to time. So present versus ancient samples. We also see some structure comparing high altitude and low altitude, and also some geographic structure, which is individuals that come from northern part of the Andes versus the ones from southern part of the Andes. We also did something similar with that, but also including populations from Europe and Africa and Asia, just to see if there is some sort of contamination. We estimate contamination separately, but we also double check doing this type of analysis, and everything suggests that those are actually ancient Native American individuals. So we have the data. We are still generating some new sequences to improve the depth of coverage. And meanwhile, we are checking or benchmarking the methods that are available in the literature to infer natural selection with time series data, in this case with ancient DNA. And this benchmarking is being done by my colleague, Lucas Anchieri at the University of Lausanne. And basically what he does is that he simulates a population evolving through time. There are many loci in this population. Some are neutral, some are deleterious, and so on. And then he has this allele frequency trajectories. And based on that, he samples this population. Here he has 20 points in the past, one in the present and 19 in the past. And each one with a sample size of 20 individuals. And here are the results for 3 methods that are described here on the right-hand side. And the red star is the actual selection coefficient that we simulated. And what we can see here is that there is only one method that performs well, and only when natural selection is adaptive, so it's positive selection. This looks okay, so we could use this method. The problem is that those sample sizes are very large. We don't have that 20 individuals per time point in the past. We have less than that usually. This is more the results comparing, using a scenario that is more comparable, like more similar to what we have with the ancient DNA. And when we do that, there is no method actually amongst those 3 that performs really well. We are looking into other methods. We are also trying to develop our own approaches to study natural selection with this time series data. And I'm expanding this research. This I started as a postdoc when I was in Switzerland, but I'm expanding that into the study of response against pathogens using in my own lab. So we are focusing on the Yersinia pestis, which is the plague in Europe, and also tuberculosis in Peru. So I don't have results for that. We actually received our first batch of data from a European population, and we are very curious to see what we're going to find. We are initially screening for pathogens to understand what is the paleopathology of those samples, right? So what diseases affect those populations and so on. But very early stages, I have no results or data yet. With that, I would like to thank everyone who is watching, the organizers of the speaker series, in particular, Ricardo, for inviting me. I also would like to thank my collaborators. Their list is here. There are many, and also the people in my lab. And I have my contact info here in the bottom. So please feel free to reach out to me if you have any suggestions or questions. And I would like to, I think we still have time. I have about 15 minutes, right, for questions and discussion. I hope everyone was able to hear me. I know there was some issues with the audio and with the video, but I hope it worked for most of you. Thank you.
Yeah, so I just wanted to thank Carlos Eduardo Guerra Amorim again. Thank you very much for coming. So the floor is open for questions. I also wanted to note that this is actually live from Brazil. Is that correct?
No, I came back last week. I'm in the US now. Yeah.
I thought he was going to be in Brazil too. But yeah, we have quite a bit of time for questions. So feel free. And here's a quick question. For your DFE model, were you using specific genes or was it a whole genome sequencing model?
So that's a very good question, because this is only for the whole genome. We haven't, so the whole exome specifically, we haven't looked into specific genes. We do want to look at specific genes that we believe are related to artificial selection, so, such as like morphological traits or behavioral traits, maybe immunological traits as well, and diet. But I haven't been able to get a good amount of data when I subset to this data set. So I'm not sure how I will proceed with that. But thanks for the question.
So the other question tag along with that is, have you used any of these models like DFE to look at like, disease models like malaria? So malaria, for instance, it's well known that a specific genotype corresponds to sickle cell anemia. However, if you're heterozygous or dominant, you have resistance to malaria. So that might be a selection pressure that might be interesting.
Yeah, so that would be great. The problem with our model is that we assume all mutations are additive, and we only look at negative selection, at least for this work with the dogs. The method is being modified to also incorporate different to incorporate positive selection and also other dominance values, right? So, but right now we are only looking at additive and negative selection, so we couldn't look at something like the sickle cell anemia because of the because of the advantage of the heterozygotes and so on. It's a different dominance pattern.
So any interest in looking at historical like diseases and plagues? I know you're looking at the Black Plague, right? But there's also the Spanish flu, the yellow fever.
That would be very interesting. My issue with that, I narrowed down to tuberculosis and the plague because of the availability of data for tuberculosis in South America. There's a lot of research going on with Peruvian populations, and we know a lot about the pathogen and also about the humans that inhabit the region. For Europe, the reason I focus on Yersinia pestis is because I believed it's just a guess actually, so I needed to get some selective pressure that I thought it was very strong, and that's why I chose Yersinia pestis, because the plague is known to have killed a lot of people during the Middle Ages, and there are several outbreaks. There is one in the beginning and one in the end of the Middle Ages, and there's also a prehistoric one. So at least in Europe, we know of 3 big outbreaks. So I think my idea is to try to see how humans responded in those 3 different episodes, and what are the evolutionary gains that the pathogen gained in those 3 episodes.
So one of the things that Rose studies are like chronic diseases as well as like cardiometabolic disorders. and like aging type disorders. Have you looked at chronic diseases under these models? And like, for instance, you might have a selective pressure that we evolved against in the short term, like immediate things like infections, that might have a long-term cost for fitness on the long term as we age.
That's very, very interesting. I'm currently not looking into that, but we had a small paper a while ago that we wrote about cholesterol transporters, and we were able to associate currently a deleterious mutation that is associated with high levels of cholesterol in a lot of cardiovascular diseases. It seems to have been selected in the past, and we can associate that with a period of the people of the Americas where diet was very poor in animal fat. So it was mostly based on maize, corn. So we do see something like that we saw something like that in that study where but currently I'm not looking into this type of, I would say, evolutionary mismatch, or they also call the thrifty genotype hypothesis. So I'm not looking into that currently, but it's very interesting, and I would like to look in the future for sure.
Like I understand why you did dogs. Dogs have a very well documented selection pressure and good records on their lineage. Have you any interest in looking at other animals or other mammals that have like spread out throughout the globe, like bats, for instance, or any other ones come to mind?
Yeah, that's we are actually doing that. A colleague of mine, Mei Shi Lin, she's a PhD student. She's about to defend her PhD at UCLA, and we are collaborating in this project. It's one of her main projects of her thesis. She's looking at the DFE across every mammal that she was able to find polymorphism data. So I think she has about 10 to 15 mammals that are animals overall. So we have mosquito, we have Drosophila, we have one bird, we have different mammals. We have also the vaquita, which is it looks like a dolphin. I forgot the name. It's like a vaquita. It's an animal that is really endangered animal. So we have all these different animals in different environments under different types of selection effects. And what we observe is that the DFE is more related when the species is less divergent. So that's also what other people have been finding. And apparently, it does look like the DFE has not a lot of fast response to the environment.
That's very interesting. So any more questions before we sign off? But thank you again. Oh, we have a question.
I think I have a question, yeah. So the question is tangentially related to your research project with ancient DNA. Do you have thoughts to share on what ethical considerations researchers should think about when using ancient DNA? Yeah, thanks for the question. I'm still learning with my colleagues who are doing research on ethics with ancient DNA. But what is a consensus right now is that we should make a big effort to identify who are the populations that are descendants from these populations that we are studying. And we should collaborate with them, or at least have to be in touch with them to inform what type of results, what type of analysis we want to do, what are the implications of our results, if our results are going to have some social or political implications for them. So all those are some of the ethical considerations. There's also the issue with sample destruction. So those individuals that we study are the ancestors of present-day people? Why can we actually just destroy their skull, destroy their teeth? So we are trying to develop like minimally invasive techniques to extract DNA, to pulverize the part of the samples, right, to extract DNA and so on. So those are some of the ethical considerations. There's been a lot of the very heated discussion, let's say, in the past year and the past 12 months about that. And it's very, the field is changing a lot in adapting very fast. And thanks for the question.
All right, thank you again. Pleasure, and I think we'll sign off. Take care, and great to see you.
Thank you so much.